Hi everyone, today we're going to go over chapter 13.3 through 13.5 of your textbook, which reviews dihybrid crosses and more complicated forms of inheritance. But before we get into all of that, we need to make sure we understand the term dihybrid and how that compares to the term monohybrid. So monohybrid tracks when we're tracking a inheritance of a single trait from a single gene from parent to child. So it's you tracking what's the chances of a given seed color being passed from one parent to a child. So the chances of big Y and little y and little y parent producing a little y child. Okay? Dihybrid cross is when you're tracking the simultaneous inheritance of two different genes. Generally speaking, those two different genes will also produce or follow two very different characteristics. So what's the chances of you inheriting a certain seed color from one gene and a certain seed shape from a different gene? So in this case, instead of tracking big Y versus big Y, we're tracking between the parents big Y, big R, little y, little r. Each of these letters represents a different gene being passed on from parent to child. The R gene, which controls the shape, round or wrinkled, and the Y gene the gene we designated Y being the gene that's related to color. We're tracking the probability of both color and shape being passed, a certain color and shape being passed on to the child. So the cross that you see on the screen right now, this is actually a classic cross that was looked at by Mendel in his experiment and what led him to the conclusions he gained from dihybrid crosses. And in this case, what he did was he bred true breeding, meaning homozygous yellow and round plants with homozygous green and wrinkled plants. And he made completely heterozygous generation, and that's how he determined yellow and round were the dominant alleles for each of those separate genes, the yellow gene, the color gene, and the shape gene for round. And then he crossed these plants, and then he found this ratio of yellow to yellow wrinkled to green wrinkled plants. And this led him to a question because he knew, based on the original parents here, that the children here, they had to have gotten their yellow and round gene from one parent and their green and wrinkled gene from another parent. So he went, wait, so how are these two genes inherited? Does it matter which of the alleles you got from one parent? Does that actually matter in future inheritance and what you pass on to your next generation? And what he did was he looked at it. He knew you had to have gotten the yellow with the round allele from one parent and the green with the wrinkled allele from the other parent. So does it matter that came from a given parent, or are the alleles that you got for color and shape, are they just going to be passed on to the child regardless of how they originally were inherited from that parent's original, like, previous generation? So in this case, he did some math and some calculations. So this is the hypothesis for if it does matter that the yellow was inherited with the round allele from the original parent or the original generation, and that the green was inherited with the wrinkled. And if it did matter, then he should have expected the children to be all yellow and round, or at least a majority of them, with only 1/4 being wrinkled and green, based on the idea that you had to have gotten either option based on what came from the parents, or the original parents of each of these individuals that are crossing with one another. But that wasn't the case. He got 9, 3, 3, 1. This doesn't match that ratio. So then he tried it where it doesn't matter which allele came from which parent. That doesn't affect whether you get the round or wrinkled or the yellow or green. It doesn't matter. And when he did that cross here, where it didn't matter which color you got and which shape allele you ended up with, he found out that he got the appropriate ratios. It matched the ratios that he was getting experimentally with this model, which means that when this parent makes a gamete and they pass on one of their two copies of the allele to the child, if they pass on a yellow allele, that does not affect the chances that they pass on a round or a wrinkled allele to that gamete. Okay? So that means that when you make a prediction for a parent, you actually have to come up with every potential combination that that parent can produce in their gametes when you're producing a Punnett square prediction of what might happen in the offspring. That's a little confusing, so we'll get into some laws that kind of explain how Mendel's conclusions, and then we'll go over some examples. So Mendel, between his experiments, comes up with these two laws of heredity. We already talked about the law of segregation, which affects how alleles segregate into separate gametes, and we just go, it doesn't matter. It's just random which of your two alleles that you have for a gene and which gamete it ends up in. All right, that explains monohybrid crosses. Then we also have a second new law based on what he saw in the dihybrid cross, which is a law of independent assortment. So in this case, that means that when you're tracking more than one gene, so that's for a dihybrid cross, or it could be for a cross that involves more than one gene. So if you're tracking 3 genes, that would be a trihybrid cross. Or if you're tracking 4 or 5 genes, in that case, you can come up with the word like pentahybrid cross and stuff. But usually we just call that multigenic crosses. But in any case, when you're tracking more than one gene, they're going to sort into gametes independently. There is an exception called linked genes, and we'll get into that. But what does he mean by this? So let's just recap law of segregation. So the law of segregation, just reminding you, is that getting either allele into the final gamete is random. This picture is a perfect example of it, but let's just say you have a big A allele and a little A allele. Whether the big A allele ends up in this gamete or the little A allele ends up in this gamete is random. And because it's random, when you do your monohybrid crosses, you have to account for all possible gametes for that each parent will produce. So if this dad is heterozygous, you have to show, using your Punnett square, that there's a 50% chance he'll pass on the big H gene and a 50% chance he'll pass on the little H gene. For the mom, because she's big H, you're showing that there's a 50% chance she'll pass on this big H gene and a 50% chance she'll pass on this big H gene, which is basically just a 100% chance of her passing on big H. But either way, the Punnett square is just showing you the probability that those parents will pass on their genes to the kid, and it's random which allele that the kid ends up with, which then leads us to the Punnett square. Okay? The law of independent assortment tracks the fact that it doesn't matter that when you have 2 separate genes, it doesn't matter which gene you inherit. Most of the time, the other gene isn't going to be affected by it. So if you get the big A allele for this gene instead of the little A allele, it's not going to affect whether you get the big B or little b allele for the second gene. You make all random possible combinations. And this can happen if they're on different chromosomes, but this can even happen if they're on the same chromosomes, especially when they're far apart, because of crossing over pretty much guarantees that you're going to get random mixing and matching between the 2 chromosomes in the kids. So you still are going to have a pretty even split of all potential combinations in the final child, regardless if it's 2 genes you're tracking, 3 genes you're tracking, or more than that. Okay? That's all the law of independent assortment means. What that functionally means is when you're doing your problem, you have to basically, whenever you get the genotype of a parent, you have to come up with every possible combo they'll make up. There is an exception, and this is called gene linkage. This occurs when genes are on the same chromosome and they're very close together, which means they'll usually be inherited together. They rarely cross over and mix and match. What that means is, as we saw in the previous one, you could have random mixing and matching, and it's pretty even, especially when they're far apart. But when they're very close together, it's a lot more rare because the chances of synapsing happening exactly in this narrow space between these 2 genes is a lot lower. It's not impossible, but it's a lot lower versus unlinked genes, which follow independent assortment, meaning random mixing and matching. Now, we're not going to worry ourselves about linked genes. We should just be aware that it's a possibility. But as far as this class concerns, bio honors concerned, we're just going to follow, when we do our calculations and do our problem solving, we're just going to assume that everything is unlinked, and that's how we're going to treat any dihybrid multi-gene crosses in our class. We're going to treat them as unlinked genes, and what that means is our problem solving strategy is just simply come up with every possible option that the parent will make and then do the math. So how do we do the math? And by the way, all these laws are based on meiosis, okay? So what that means is the reason why it's random whether you get big Y or little y in this allele has everything to do with how it lined up or in this gamete has everything to do with how it lined up during metaphase 1. And the reason why you get big R to combine with little y or big R to combine with big Y, again, has everything to do with how they randomly aligned up at metaphase 1. All right? So just be aware that all those Mendel laws that affect genetics essentially is due to what we learned about originally with meiosis. But either way, let's go into how we do these problem solvings. So it's very similar to our monohybrid Punnett squares, but it gets messy because you have to come up with every potential allelic combination that the parent may pass on. So if the parent's big A, big B, big C, you have to come up with every possible combo of single allele mixings that they could have for each gene. So you might have big A, big B, big C, big A, big B, little c passed on, big A, little b, big C passed on, and you just have to come up with every potential combo this parent may pass on. If the parent is dihybrid, then you have to come up with every potential combo that this parent may pass on. Really carefully look at the crosses in the Punnett square and note that whenever you have different genes, they're designated by different letters. So this designates, the A letter designates one gene, the B letter designates a second gene, the C designates a third gene. Also notice in the Punnett square, when we write the genotypes out of the crosses, we always want to make sure we pair up the alleles from the same gene together and the alleles from the other genes together. So because each of these genotypes are read separately. This is round, this is round, this is wrinkled, this is yellow, this is green. So we want to make sure the alleles for the separate genes are paired when they show up, when we write it out in the combined dihybrid or trihybrid cross or whatever. And again, same rules as before, we want to make sure the letter that is going to be the phenotype shows up first. So the capital letter always goes in the front of my pairs within the gene that you're looking at. That's going to matter when you do your Punnett squares. Don't do weird stuff, otherwise you're going to get really messed up Punnett squares. Okay, so anyways, you have your genotypes for the parents, and what you want to do is come up with every possible combination of the Y mixing with the R. One way to track it is do what we call FOIL, so just the first letter of each genotype, the outer letters of each genotype, the inner letters of each genotype, the last letters of each genotype. That's one way to track it. I just do it by memory, but some students have a hard time tracking everything, so just work through each letter systematically, and then you'll get all possible combos. Once you get all possible combos, just like before, you write them on each side of the Punnett square. You combine the letters from the same genotype together, making sure the dominant allele is always first. And then after that, you read it individually. You have a yellow and round individual here. You have a yellow and wrinkled individual here. You have a green and round individual here. You have a green and wrinkled individual here. You'll count your Punnett square up, do your comparison for the ratios, and then this is out of 16. And then you're going to reduce all your numbers if they all have a common number. So in this case, it's 6, 6, 2, 2. All of them are divisible by 2. So now the ratio is 3 yellow round to 3 yellow wrinkled to 1 green round to 1 green wrinkled. Or if you want to make them fractions, 3/8, 3/8, 1/8, 1/8. Okay? So it's just simply like before, but it's more complicated by the fact that your Punnett square is a lot bigger because you have a lot larger variety of possible combos. And just a heads up, remember, each side of your Punnett square is just all potential combos from a parent. So you could, in theory, have a smaller Punnett square because notice for the second parent here, you only actually have two options, big Y, little r, little y, little r. So you could actually have a lot shorter Punnett square and still get the same answer as you had before. See this one versus this one. It's the same answer. You can also do math. You don't have to do a massive Punnett square, but this is only going to apply to certain situations. And let me go over those situations. So how does the math work? So we have two laws of probability called the rule of multiplication and the rule of addition. That actually guides your Punnett squares. So your Punnett squares are just an example of probability. It may not seem like it, but it is. Why? Well, you have a half chance of getting the big P from this parent and a half chance of getting the big P from this parent. Half times half is 1/4. Oh, this is 1 out of 4 squares. So the Punnett square is just a tool of probability. the chance of getting little p is the same. You have a 1/2 chance of getting little p from this parent, a 1/2 chance of getting little p from this parent. It's just 1/4. 1/4, 1/2 times 1/2 is 1/4. So this is just modeling probability. That's why we do the Punnett square. It's just a clean way of doing probability. And also the chances of getting heterozygote here and here, it's a 1/4 chances this way of getting the big P from the mom, and a 1/4 chance of getting this heterozygotes by getting a big P from your dad. And you add those together, and that's why it's 2 out of 4 squares, or 1/2. That's a little bit complicated, but basically we're coming down to these two rules. The law of multiplication, or the rule of multiplication, is just when you want two events to happen at the same time, you can actually just calculate those two events in a more simple way and then multiply the chances together, and then you'll get the right probability. The law or rule of addition is just when you see a problem asks you or, meaning what's the chance of this or this happening, you can calculate those two separate probabilities and then just add them together and you'll get the answer. So what does that mean? Well, in terms of multiplication, this is going to be huge because it's going to prevent you from having to do massive Punnett squares. So let's say you need to do a cross between these two parents. This would be a nightmare of a Punnett square. But let's say all you need to do is calculate what is the chances of making this child. Big A, little b, little c, big D, big E, big F, big F. Instead of doing a massive Punnett square, just calculate the individual chances of making this A genotype, this B genotype, this C genotype, this D genotype, and that E genotype, and that F genotype. So calculate the individual probabilities of each of those genotypes. And after you calculate it, instead of making a massive Punnett square, because all these events have to happen simultaneously for this individual to be made, just multiply all these genotypes together. And instead of making a massive Punnett square, you just solved this problem using math. And then my answer is 1 out of 64 instead of making a giant Punnett square. That's how you use the rule of multiplication. And you can apply this to dihybrid crosses. So for a dihybrid cross, just like before, we have these same genotypes, but instead of making that more complicated Punnett square, instead, all I have to do is calculate, if I just want to ask the question, what's the chances of making a yellow and round seeded plant? I just calculate what's the chances of making a yellow seeded plant versus what's the chances of making a round seeded plant, and then I just multiply them, and I get 3/8 are yellow and round. So instead of doing this massive complicated Punnett square to find out 3 out of 8 are yellow and round, I just do the math for each part individually and multiply them together, and I get the same answer. So that's a lot easier than doing a complicated Punnett square for some people. All right, the or question can be a bit more complicated. So in this case, what's the chances of producing a green or wrinkled plant? In this case, you can't just calculate what's the chance of making a green plant and what's the chances of making a wrinkled plant and add them together, because green and wrinkled overlap sometimes, so you might double count them. So unfortunately here, the easier solution, you can do it through math. You just have to calculate the chances of being green and wrinkled, the chances of being green and smooth, and the chances of being yellow wrinkled, meaning write down every single possible phenotypes and then figure out which phenotypes apply for this problem and then individually calculate them. But for more complicated problems, honestly, just the more complicated Punnett square is a lot simpler, and then you just add the things that help out that would count together and then get the right answer. So in some situations, you're better off with a Punnett square, and you're better off with the Punnett squares when you're asked to provide the genotypes or phenotype ratios of everything or a lot of things. But in situations where you're just asked what's the chances of this specific type of child happening, just use the rule of multiplication. It's so much faster in some cases than having to make a massive Punnett square for a dihybrid, trihybrid, or any multigenic cross. So to put it in perspective, Mendel got incredibly lucky because the plant that he was studying had actually very simple genetics. Basically, pretty much all the genes he looked at were dominant recessive, meaning that as long as you had one allele that was dominant, it would show that trait. So the heterozygotes were indistinguishable from the homozygous dominant plants. This is different than what actually happens in all genes. So there are more complicated forms of genetic inheritance and genetic patterns. So in this case, there are actually other versions of monohybrid cross where the heterozygotes actually look different than the homozygous dominant or homozygous recessive alleles. So we have two versions of that different heterozygotes production. So in this case, we have incomplete dominance where the heterozygotes looks different, but the reason why it looks different is because it shows an intermediate blended phenotype due to dose. What dose means is just because it's incomplete dominance, it means that the dominant phenotype, in this case red, only is truly present in its pure strongest form when there's both alleles are dominant, meaning both alleles have the red allele protein information. And when you only have one of your two alleles have that red allele protein information, then what ends up happening is you kind of show a partial expression of that dominant trait, incomplete dominance, instead of that full trait. So you ended up with a blended phenotype in that heterozygotes, while the one with the homozygous recessive trait just has an absence. Now, sometimes because the heterozygotes is a medium trait, and it's sometimes difficult to tell which one's the dominant trait versus which one's the recessive trait, sometimes people get around that by just designating each of the homozygotes as a different letter. So this is the red flowers are the R allele and the white flowers are the W allele, and then the heterozygotes is RW. Okay? But it doesn't really matter whether you choose big letter and little letter to show the homozygotes or you choose different letters for these incomplete or codominant traits. It'll still work. Your problem solving will still work. Now, codominant traits are actually codominant, meaning it's showing that these two different alleles that you have both want to express themselves, and if they show up at the same time, they're both going to fight and simultaneously show up. So the trait is not blended. So you, instead of pink here, if the red and white alleles are both codominant alleles, it means the white expresses a protein that wants to show, and the red allele expresses a protein that shows. So the blended version, the heterozygote, isn't exactly a clean, smooth mixture. It's actually patches of red and white, both battling to show up in the final child. So codominant and incomplete dominance, in terms of calculating and problem solving, it's actually going to be very similar because their heterozygotes look different. But the reason for why the heterozygotes look different are for different reasons, and the outcome of the heterozygotes is slightly different in that codominant will always show up if both alleles show up, while incomplete dominance shows this kind of blend because the dominant allele isn't super strong. All right. So in terms of solving monohybrid inheritance for these guys, this is just kind of a summary of what I went over with dominant, incomplete dominance, and codominance, and how and the reasons why it might end up blended versus it ends up showing up simultaneously versus dominant being dominant when even in the heterozygotes. And then there is a third version of monohybrid inheritance called X-linked inheritance, but you aren't going to learn about that in this lecture. You'll learn about this in the next lecture. But anyways, in terms of solving the Punnett square, it's actually still pretty much the same as before. The only difference is, just remember, the heterozygotes look completely different than the homozygotes. So if you have a homozygous red flower mixed with a homozygous white flower, the Punnett square is basically the same setup, but because the outcome is heterozygote, instead of having all red flowers, this time you have all pink flowers because that's the blended trait in this question. Okay? Ditto over here. If you have the two pink flowers crossed together, you have the pink alleles R and W over here and the pink alleles R and W over here. And then with the children, instead of having what you would normally expect, meaning 3 dominant phenotypes and 1 recessive phenotype, because the heterozygotes look different, you have 1 all homozygous red, 1 all homozygous white, and then 2 out of 4 being pink, or 50%, 25%, 25%, in terms of your ratios and probabilities. All right, it's just different in that you have to account for the fact that the heterozygotes are going to have a different phenotype, But otherwise, solving the problem is very similar to how you solve the problems in monohybrid crosses. Here's another example with cats, where you learn that the long-tailed cat is crossed with a short-tailed cat. And in this case here, because the question didn't tell you there was a medium one, but you knew that these were the individual genotypes, and because you know the genotype of short is little h, and the genotype for long is big H, then you should know in this case here, because it's incomplete dominant, the heterozygous is a blend, so you can hypothesize it's medium. And then when you do the cross, then you'll see that, oh yeah, all of them are going to be medium. That's it. Quickly over a classic form of codominance, which is calculating the chances of passing on type A, type B, and type O blood. All right. So the blood type groups are actually based on the antigens on the surface of your blood cells. So if you're type A, you actually make a special marker on the surface of your red blood cell. And because that red blood cell marker is type A, it means that you are not training your body to ignore the type B blood. So if you're type A and you receive type B blood, you're going to attack it. If you're a type B blood, you're going to express, because you're dominant, you're going to express a type B protein on your surface, which means your body learns to ignore type B proteins, but it's going to also learn to attack any type A proteins. So type B people cannot receive any blood from anyone with a type A protein. Okay? If you are AB, that means you express both A and B surface markers, which means no one else can get your blood. Because a type A person would attack the B marker, a type B person would attack the A marker. But what that does mean is if you're type AB, you don't attack any other blood types because you have all the markers. So you can receive blood from anyone else. But if you're type O, that's recessive, that means you have an absence of antigen, so you don't make any proteins. And in this case, your blood can be given to anyone else if you're type O, but you can't accept blood from anyone else because your body will attack it. Now, these are the genotypes that lead to these different blood types, because A is produced by a dominant allele, B is produced by a dominant allele, and O is just a recessive allele. It means that if you have type O, it's just because you lack any other dominant alleles. So some people write the genotypes this way to just show that, hey, this is dominant and this is recessive, but you can also just write it in terms of the letter of blood type that it's associated with. Just remember O is recessive. That's it. So in this case, you just treat this normal like any other cross. And just a heads up, this picture kind of shows you how the blood types work and why the antibodies will lead to dangerous clumping and attacks, right? Type B blood people will recognize type A and start to attack type A blood if it receives it and cause clotting. Dangerous. Anyways, so the crossing here is you just have to use those genotypes that I showed you in the previous tables. In this case, you have type B blood, and it's heterozygous, which means you have to have an O allele, because O is the recessive allele that doesn't show. You can't have an A allele, otherwise you wouldn't be type B blood, you'd be type AB blood. All right? So anyways, we have the B allele and the O allele for both parents, and then you just do the cross. And again, you can simplify your life and just write the letters associated with each blood type here. So we have the B allele and the O allele for each parent. And then when you do the cross, you can see, the chances of a child having a B allele, either only B alleles or B and O leading to type B blood, is 3 out of 4, while the chances of making type O blood is 1 out of 4 for this cross. If you take it for another more complicated problem here, where you have a type AB parent and a heterozygous type A parent, now you have a kid that has type A blood here because they have only the A alleles, type A blood here because they have a type A allele and an O allele that doesn't do anything, so we have a type A blood here. You have a type B child here because they have a B allele and an O allele. O doesn't do anything, so it's B blood. And you have a type AB child here. So if you do the math, it's a 50% chance of making a type A child, a 25% chance of making a type AB child, and a 25% chance of making a type B child. So that's just how blood typing works. It's just the same crosses we've done, but the alleles are just a little bit funkier. All right, last thing I wanted to mention before we wrap up is just the fact that traits are not always affected by one gene. I know we've mainly only covered examples with traits affecting one gene, but in reality, life is more complicated than that. There are examples of one gene affecting more than one trait. There are also examples of a trait being affected by many genes. So, example, epistasis, one gene may completely mask another gene, and those two genes work together to show a trait or not show a trait. And then polygenic traits show a range of genes affecting and adding together to affect one trait. And then we have epigenetics, where your genes are not the only thing that affect your traits. The environment around you and how you live your life affects your traits. So here are examples of each just so you get a better idea of it. So dwarfism and gigantism are all affected by the same gene. So, and also on top of that, dwarfism isn't just about height, it also affects your heart condition and stuff like that. Polygenic traits are many genes affecting one trait. So in this case, skin color is actually affected by many genes at once, all adding up together, which is why, and all of them are incomplete dominance, which is why the number of alleles you have for each of those genes all add up together to determine how much melanin you make. And that's why we have this wide range of skin tones available in humans. You also have albinism, this example of epistasis. So you have your gene controlling how much melanin you make versus a gene controlling whether or not you can make melanin. So maybe you are supposed to make really dark skin, but because you got a gene that means you can't make melanin, now you're just albino. And then finally, you have environmental effects. So the way you live your life can determine whether your genes, even though you have the gene information, it may not be read because the way you live your life caused that gene to be closed up and wrapped tightly around the histone, so you're not using it. This is actually how animals can change their fur color in the winter, because they normally make fur color, but in the winter, the cold causes their genes to wrap up tightly, so now they're white, and guess what? They're a little bit hidden in the snow now. So, those are just some more complicated things to think about when it comes to genetic inheritance. But mainly what you want to practice from now on is just problem-solving strategies. So anyways, that wraps up today's lecture, and just move on and start practicing new problems. Bye.